And the question is, what are sort of the transitions we're going through? To introduce you to what's going on, I want to talk about a virus, a biological virus, to start. And then from this, I'll get to a more open-ended programming framework for engineering genetic material. So if you haven't seen a biological virus, this is a virus that infects not people, it's a virus that infects bacteria. There's an electron micrograph on the left, and then a schematic diagram of the particle, the virus in the middle, and then an action shot on the right. This is a physical object. It's about 100 nanometers in scale. And what it does is it carries some DNA around. The DNA encodes a program that causes bacteria to make more copies of itself. Rather than talk about this, I'll just show you a movie of one of these things. These are bacteria growing under the microscope. And as they're growing and divided, they've all been infected with a virus. And because the virus is so small, you can't see it. And then eventually the cells are destroyed. Each time the cell pops and is destroyed, that releases several hundred copies of the virus to repeat the infection process. So the key features of biological technology, in this virus is one example, is that it's a self-assembling technology. Right? I can take the bits and pieces and throw them in a soup and they make stuff. You know, I don't have to solder it together. That's pretty impressive. It operates on the nanoscale, so it's a nanotechnology, really. You don't have to read Eric Drexler and have a bunch of fantasies. You actually have a nanotechnology. It's called biology. It's a reproducing machine. So this is an object that can copy itself physically, and you can program it. You can program it via the genetic material, via DNA. So to an engineer, this is an amazing technology platform. The sad state of affairs is we're really bad at doing the engineering of it. So just to motivate this example from some external perspectives, this is a world map showing chlorophyll